Hello, my name is Niall McKay and welcome to Living with PSC, a podcast about the rare disease of primary sclerosing cholangitis. Now, PSC causes the bile ducts inside and outside the liver to become scarred, narrowed and eventually blocked, and leads to liver cirrhosis. In this podcast, we aim to dispel some of the myths about the disease. Today we're talking to Dr. Espen Melum, MD, PhD with the Norwegian PSC Research Center at Oslo University Hospital, about a new discovery about the role of contaminated bile may contribute to the onset of primary sclerosing cholangitis. Welcome, Dr. Melum. You discovered antigens that activate mucosal-associated T or MAIT cells that are present in the bile in a number of PSC patients that you studied. Can you tell me what a MAIT cell is and what role it's playing in the bile?
So first, thanks a lot for having me and allowing me to discuss these findings with you. So MAIT cells is a rather recently discovered subtype of T cells, which is, of course has been around all the time, but just passed around as normal T cells. What is specific about MAIT cells is that they are reacting to vitamin B metabolites instead of peptides, which is a normal action of T cells. And these, what is the smart thing of the body to react to these vitamin B metabolites is that they can only be produced by bacteria. There's no cells in our body that can produce them. So when the T cells see some of these antigens, they know that there's something foreign there. So in the terms of evolutionary perspective, this is a clever method. And what makes these cells especially interesting in the context of liver is that they're highly enriched in the liver. And as the name saying, they're mucosal associated, and the bile duct represents an extension of the mucosal surface in the bowel.
So this is kind of like a connection between the bowel and the bile, if you like.
You can say that this is what we will speculate in the next round. We had some basic discoveries in this paper, and of course we're then speculating what this can mean in the broader context, and also during both the physiology and disease, which we're investigating in terms of other projects.
And this would explain the sort of connection between why, say, 80% of PSC patients also have inflammatory bowel disease or Crohn's or colitis. Is that correct?
No, I think that's a way too long stretch. I think this is one piece of how the immunology works. And I think there would be other mechanisms that would be involved in determining the inflammatory bowel disease in PSC patients.
I see. And do you consider, is PSC, I mean, there seems to be a question as to whether PSC is autoimmune or inflammatory or both.
I think that these results are not arguing one way or the other, but my general view on it is an inflammatory disease. It doesn't fulfill the classical criteria of autoimmune diseases, and that's why I don't want to say that it's an autoimmune disease. I think in practice it doesn't matter. We know that the immune system plays a major role, and that is where we're focusing our research.
Can you explain to me, like, how do the T cells work with a bacterial contamination, or is that a separate issue?
No, these are general concepts. Of course, when there are bacteria, the cells will recognize the antigens and start up the immune response. And when we're finding that there are these specific antigens are present in bile, we can think that MAIT cells play then a role in giving the inflammatory tone, not necessarily as the initiating event, but maybe during disease course that they might be involved.
So what happens is that there is some contamination in the bile that activates the T cells that kind of overreact, if you like, and start causing scarring, which causes the strictures to be blocked, or is that an oversimplification?
No, I think it's. I don't think it's necessarily an initiating event, because what we discovered is that normally bile was thought to be sterile, and that there were only bacteria there in the setting of ongoing cholangitis. But a range of other studies, ours including that, is that there is some degree of low-grade colonization of the bile ducts, and so that's one of the reasons we went into this and see if these bacteria that are there at a low grade, whether they're containing antigens, since bacteria are producing the antigens for MAIT cells, whether the bile would also contain these antigens, and that's what we showed in this paper. Whether it's an initiating event, I doubt, but that it plays a role in either sustaining or regulating the tone of inflammation. That's at least a hypothesis that we're following.
Yeah, it's why often doctors and scientists call PSC a black box disease, isn't it? It's so complicated, you know.
Yeah, I think it's complicated. There's many aspects, but at the same time, if you look at these other inflammatory diseases, which are not that common, they would be equally complicated. So I don't think it shouldn't lead to us being skeptical about pursuing this, because yes, PSC would be difficult, but the same way would be any autoinflammatory, for instance, lung disease or kidney disease. It's the same challenges, but it's, of course, PSC is a small disease, hasn't been investigated that heavily in the previous years. That is luckily changing now.
What role do antibiotics play with the MAIT cells?
So what we showed here is we didn't do any antibiotics experiments or look at any association with antibiotics, but since these antigens are produced by bacteria, and as I said, since this synthetic pathway for antigens are not present in mammalian cells, they need to come from bacteria, and you can change the bacterial flora with antibiotics. And that's why we looked into these different bacteria that we found, and on a theoretical perspective, looked into whether they could, whether they were susceptible to antibiotics, which several of them were. So this would just mean that you can potentially, this is common knowledge because we treat patients that have a very high tendency to cholangitis. They're even on prophylactic antibiotics. So
yeah, and are there specific bacteria you're talking about, like Klebsiella pneumonia, or do you know what they are?
No, I don't think we would incriminate a specific bacteria. We were just interested in whether they were something that potentially can be modified, but still, that doesn't mean that if we modify them, that it would do anything to the disease course. And of course, there's lots of off-target effects regarding antibiotics. So I would say for antibiotics is following regular clinical guidelines and use when needed, not to sterilize the bile ducts when it's not known whether modifying this would have any effect on the clinical outcome. That's what means something in the end. The thing with this paper is that it introduces new concepts that can be tried out, as we're doing now in animal studies, and also generate hypotheses for potential future treatment.
Yeah, because it's a sort of a dilemma, isn't it, for you doctors treating PSC, is that you can influence the disease or bring down the contamination, if you like, or treat the disease somewhat with antibiotics, but then they create all sorts of other problems if you overdo that.
Exactly, which is the problems that you don't want. And this is a general thing with antibiotics. There's so much off-target effects, and of course, we're willing to accept off-target effects if it has a huge benefit. But then the benefit needs to be proven in clinical trials.
The MAIT cells activating antigens were of microbial origin. You studied 35 and you found that, say, 15 people with PSC had bacterial colonization. Now, did the control group or the other group, or not the control group, the other group, did they not have bacterial colonization in their bile?
Yes, we did find some of them there as well, and but the point was that these were not the antigens that were there. We're just giving some unspecific activation. The reason whether this is PSC specific, I don't think, but PSC patients have a lot. They get all these strictures. and also are going to maybe to repeat the DRCPs, and then you kind of open up an entry point for the bacteria. If there are patients with other diseases in the bile duct which leading to strictures and also subjected to ERCPs, then I would expect we see the same findings there.
I see. And what role would this have, say, in a transplanted patient? Because sometimes the disease can reoccur well, I think in about a third, I'm not sure if my figures are correct, but about a third of patients, PSC reoccurs after transplantation with the new liver.
This is, of course, the third is approximately our estimate as well, and this is a very interesting topic which is heavily investigated as I think this will, which is something that can really teach us about PSC in general as well. Whether these mechanisms are there, I would say if the bile ducts are colonized in the same manner in transplanted patients, I think they would play the same role. But that is purely speculation.
You do great work there at the Norwegian PSC Research Center. Is this part of a wider study that you're engaged in at the moment?
Yeah, thanks a lot. Yeah, this is. I have a research program on unconventional T cells, which involve these MAIT cells, and another cells called NKT cells, where we had parallel findings with lipid antigens that we published last year, and we're also testing out these kind of antigens more directly in animal models, and just to study more the pathophysiological process going on when they're activated.
Are the antigens also present in the mucosa in the bowel? Is that what you said originally?
Yes, correct.
Yeah. So, and do they travel to the bile, or do they occur simultaneously in the bile?
So our hypothesis is that this is caused by the low-grade colonization, and the bacteria is present in the bile. Of course, in the bowel, there's lots of bacteria, so they also have the same synthetic machinery. Luckily, the bowel wall is pretty tight, so they're not letting in all the time.
Dr. Melum, is there anything I didn't ask about your study, or anything else that you'd like to add?
No, I think I'm impressed by your interest and your insight into this. I think it's you asked a lot of very very relevant questions, and I would say I'm a little sorry to disappoint you that this doesn't have any direct consequences for the patients. But what I would like to point out that these kind of studies it helps us identify concepts for inflammation in the bile duct, which after maybe 2 or 3 other series of experiments, identify something that can be treatable down the road without us able to say what it is now.
There's a lot of research at your particular stage at the moment, which unfortunately, as somebody who has the disease, I'm not sure that it'll be ready in time for me, but I think one of the things that seems to be emerging is the prognosis or the future looks good for being able to decode this disease and perhaps treat it probably in 5 to 10 years.
I'm optimistic, and you think about these kind of studies. You never know what kind of the concepts that will drive into treatment. You cannot see that before you have started doing these studies, and which kind of will win and which concept will be the relevant one. So I think it's very important that there's a broad level of studies looking into different aspects of understanding the disease, and also in terms of like shorter term, I would hope that the type of treatment would be something that where we can slow down the disease progress to such a degree that it's not such a big problem for many patients. I think that's more realistic than being able to give one drug where you get rid of the disease.
Yeah, well, that would certainly be good news because the liver is actually very robust in a lot of ways.
Exactly.
Yeah.
Exactly, and you can, and as long as you have something ongoing which you can slow down, then patients don't need to think about it, and then it would be easier.
Yes, yeah, no, I think it's great work.
Yeah, but I think it would be. It's very. I think it's unrealistic that it's something that can get completely rid of it. But that's just my personal thinking. But the problem would be going away if you slow it so much down.
I understand. Yeah, yeah, yeah. So that it's not as active in the host, if you like, or the patient.
Exactly.
Yeah. Well, I believe that you're also hoping to attend the PSC conference online.
Yes, I will. I will be taking part in a breakout room together with some other patients. I don't know. I think it's on the Friday.
Wonderful. Well, I look forward to seeing you there, and I'm delighted. Thank you very much for your service, both to the medical from the medical side and to the PSC community. We really appreciate it.
Thanks a lot, and really good luck with the conference. I think it's great that you arrange it even online, because I know it means a lot to arrange for different patients, and I'll hopefully also have more outreach to Europe now when you're doing it online.
Wonderful. Thank you very much.
Thanks a lot.
Well, I'm afraid that's all the time we have for this week. For more information about PSC and for a list of people and topics we'll be covering in the near future, please go to www.pscpartners.org. That's www.pscpartners.org. PSC Partners is a non-profit organisation dedicated to providing education and support to primary sclerosing cholangitis patients. There is information and resources on the website. There is a conference once a year. There is a private Facebook group that you can also join. Also, please join our patient registry. This is an anonymous database that provides research support to the medical community who are researching this disease. Thank you very much for listening.